those of you. So you're okay, Lindsay, if we record?
Yep, not a problem.
For those seminars which are recorded, you can go to the JHU Center, NIMH Center for Novel Therapeutics of HIV-associated neurocognitive disorders. If we get refunded, we are going to shorten that title. And you'll find the recorded seminars there. They're also going to be disseminated to the other centers. So UCSD, Nebraska, and Temple will also have those seminars. So with that, I will pass it over to Kelly.
Thanks, Norm. It is a great pleasure for me to introduce Dr. Lindsey Festa today. Lindsay got her bachelor's degree at Haverford and then pursued a master's followed by a PhD with Olympia Meucci, one of our dear colleagues at Drexel University College of Medicine. She made seminal discoveries during her master's work looking at the novel roles of ferritin heavy chain in HIV-associated neurocognitive disorder. And then during her PhD, she looked at the role of CXCL12 as a neuroprotective agent in the context of HIV infection in the CNS. And so she had a really strong background in neuroprotection, HIV in the CNS before joining my laboratory and my collaborator and co-mentor for Lindsay, Judy Grinspan. So together, we have been working with Lindsay where she has very quickly become not just the student, but the master of all things, as is her normal way from what I have understood in terms of understanding the role of antiretrovirals, HIV infection in the context and their impact on oligodendrocyte biology. And so today she's going to share a story she has been working on looking at the effect of antiretrovirals on oligodendrocyte maturation, which we think has interesting implications both for considering what patients at both ends of the age continuum, adolescents as well as aging patients taking antiretroviral therapy. And so some interesting implications for the clinics, as well as I think revealing some novel biologic processes that are important in oligodendrocytes, making them uniquely vulnerable to various forms of insult. And so really great to, I'm really happy and excited to introduce Lindsay today and her cat, Oreo.
Yes.
Take it away, Lindsay.
All right, awesome. So thank you very much, Kelly, for that very kind introduction. And thank you to the organizers for giving me the opportunity to provide an update on the work that I actually presented earlier this year in January. And as Kelly mentioned, one of the overarching goals of the project in the lab is to examine how antiretrovirals used to treat HIV infection can contribute to the persistence of white matter abnormalities and cognitive deficits in these patients. So I know that everyone on this call is obviously very familiar with antiretroviral therapy or ART, but just to really give a brief introduction, we know that it's been a real game changer in terms of how HIV infection is treated and controlled. It's not a cure, but it has led to a significant reduction in age-related deaths in the United States and globally, despite the number of HIV infections continuing to climb. And this is, I think, very nicely graphed here, where we can see that ART was introduced in 1995, and there was really this sharp decline in deaths associated with AIDS. So in terms of how antiretroviral therapy is able to achieve this, it reduces the amount of HIV in the body by preventing the new infection of cells, which in turn then reduces the risk of HIV transmission. It also protects the immune system by allowing for CD4 positive T cell recovery and prevents the progression to AIDS. And so ART is able to prevent new infections by targeting different aspects of the HIV replication cycle. And so typical frontline therapy is currently an integrase inhibitor, and the one of the ones that is in the frontline therapy is bictegravir, which I'll be talking about today. And it's also usually prescribed with 2 nucleoside reverse transcriptase inhibitors, such as entrectinib and tenofovir alafenamide. And ART has really done a fantastic job of decreasing peripheral viral load in HIV patients, slowing the progression of HIV. However, viral reservoirs and chronic inflammation are still found in the central nervous system, leading to cognitive disorders. So HIV-associated neurocognitive disorders, collectively known as HAND, as I'm sure we're all familiar with, comprise the motor, sensory, and cognitive deficits that are observed in HIV-positive patients. And Typically, these patients are categorized into one of three subtypes, ranging in severity from asymptomatic neurocognitive impairment, mild neurocognitive disorder, or HIV-associated dementia. And it's important to note that despite the introduction of antiretroviral therapy, the overall incidence of hand has not changed, though the extent of the severity has. And this is very nicely shown here on these pie graphs over here, where prior to antiretroviral therapy, we have a pretty large portion of individuals with hand having the most severe form. And then we can see in the CART era that this incident of the most severe form of hand has decreased, but we still have a large portion of individuals being diagnosed with either mild neurocognitive disorder or asymptomatic neurocognitive impairment. So in terms of trying to understand why we're still seeing this persistence of neurocognitive disorders in these patients who are on suppressive antiretroviral therapy, looking pathologically, one of the most persistent neuropathological changes observed in these patients are myelin abnormalities. And it's really become increasingly clear that viral suppression alone is not sufficient to mitigate neurocognitive impairment, and that the persistence of this white matter dysfunction may be due to the antiretrovirals themselves. There have been a number of human studies demonstrating the continued presence of white matter abnormalities in patients treated with ART. And this includes a thinning of the corpus callosum. And this, I think, is really shown nicely in this figure on the right, where we're looking at individuals who are HIV positive and also on antiretroviral therapy. We can clearly see that they have a thinner corpus callosum compared to an individual who is HIV negative. Additionally, the duration of CART has been correlated to the amount of white matter attenuation. And importantly, looking at transcriptome analysis, transcripts that are critical to the regulation of myelination remain dysregulated in those who have hand and are also on ART. And so before kind of going a little bit more into my data, it's really important for us to briefly, obviously, introduce the myelin-producing cells of the CNS, which are the oligodendrocytes. And so myelin, which makes up white matter, is this multi-layered lipid-rich membrane that's formed from the extensions by mature oligodendrocytes. And so looking at this EM image on the left, we can see these very nice concentric wrappings around this axon in the middle. So this is formed by mature oligodendrocytes. And mature oligodendrocytes are the only cells in the CNS that produce myelin, and they're generated from a self-renewing pool of oligodendrocyte precursor cells, also known as OPCs, which are shown here on the right. And this differentiation from OPC into mature oligodendrocytes follows a really well-characterized progression. So OPCs will transition into immature oligodendrocytes and begin to extend actin filaments to elaborate these processes, as shown here. And then upon following contact with neurons, these maturing cells further extend these processes, upregulate the expression of myelin proteins, and ultimately ensheathe neuronal axons with the lipid and protein-based myelin membranes. And when we typically think about oligodendrocytes and their roles in the CNS, the first thing that comes to mind is that they allow for saltatory conduction along axons. But it's really important to note that that's not the only thing that they do in the CNS. They actually play numerous critical roles in homeostatic function within the adult CNS. This includes myelin remodeling in response to learning and plasticity. They also secrete extracellular matrix molecules that actually helps trigger the formation of internodes, as well as also for nodal maturation and maintenance. Additionally, they also can alter neuronal transmission by modulating excitability and neurotransmitter release. And they also provide lactate to axons via the periaxonal space here. And they also remove potassium ions from that space. And so what I really want to emphasize is that these cells are really positioned to influence the health and activity of neuronal circuits, and injury to these cells can have a deleterious impact on neuronal activity and cognitive function. And our lab has shown that these various classes of antiretrovirals, including protease inhibitors, such as ritonavir, saquinavir, and darunavir, as well as one of the newer classes of drugs, the integrase inhibitors, such as bictegravir, can actually negatively impact oligodendrocyte maturation. Interestingly, though, another integrase inhibitor, raltegravir, actually had no impact on oligodendrocyte maturation function. So as these new therapeutics are becoming available for HIV infection, This really creates a constant moving target for the lab to try and understand if they alter oligodendrocyte function. So the drug that I'll be talking about today is bictegravir, which, as I mentioned, is part of the recommended frontline therapy. It's an integrase transfer inhibitor. It was, until earlier this year, the most recently FDA-approved integrase inhibitor. That now belongs to bictegravir. This was approved in 2018, so we really don't have any information as to how or if and how this drug could potentially impact oligodendrocyte maturation function. That's really what the goal of this project is. So in terms of our overall hypothesis, we are hypothesizing that select antiretrovirals, including bictegravir, negatively impact oligodendrocytes via this inhibition of differentiation and contribute to CNS dysfunction in ART-treated HIV-positive patients. So the first question we wanted to ask is, obviously, does bictegravir have any impact on oligodendrocyte maturation? So just to briefly introduce the OPC differentiation paradigm that I'll be discussing and using for my in vitro studies, we take P0 to P4 rat pups and dissect out and isolate out the cortices. And this contains a mix of cells, including OPCs, astrocytes, and microglia. We then allow them to reach approximately 95% confluency. And then, using a modified shake-off protocol, we are able to isolate out pure primary rat OPCs, typically about 95% pure. We then allow them to proliferate and grow until about 70% confluency. And following this, we then remove them from their growth media and put them in this well-defined differentiation media. And what this does is this actually stops OPC proliferation and then drives them into differentiation into mature oligodendrocytes. And it's during the 72-hour differentiation media treatment that we then are going to be adding either DMSO as our vehicle or various concentrations of bictegravir. After those 72 hours, we then can probe for either using immunocytochemistry or immunoblot. And in terms of the concentrations of bictegravir that we're using, we base this on the plasma Cmax that we see in these patients. And so the medium concentration of bictegravir reflects the plasma Cmax seen in patients. The high is 3 times that, and the low is 1/10 of that plasma Cmax. So looking at immunocytochemistry, looking at both an immature oligodendrocyte marker, which is Gal-C in this case, and then also looking at mature oligodendrocytes, which is going to be marked with MBP positive cells. What we saw was that there was a decrease in both immature and mature oligodendrocytes across all 3 concentrations of bictegravir that we treated, with no change in overall number of DAPI positive cells, suggesting that bictegravir in this case is inhibiting oligodendrocyte maturation. Additionally, when we looked at the number of precursor cells following bictegravir treatment, so we did not see any changes at the low and medium concentrations of bictegravir. However, we did see a reduction at the highest dose, a very slight reduction. So this suggests that it isn't the fact that we're not having this maturation because the OPCs are dying. And here we're just including a positive control where cells are kept in growth media. So that's why we have a larger percentage of OPC positive cells there. So now that we see this inhibition of differentiation looking at ICC, we also want to examine the expression of these mature myelin proteins within these differentiated oligodendrocyte cultures. And interestingly, we actually only saw a significant decrease in the expression of both MBP and PLP, which is another mature myelin protein, only at the highest concentration of bictegravir. We did see a slight reduction at the medium concentration of bictegravir with PLP, but it was not significant. So taking these 2 things together, it does appear that bictegravir is indeed inhibiting oligodendrocyte maturation and is altering myelin protein expression only at the highest concentration. So obviously, observing this, I really want to then try to understand what mechanism is potentially underlying this inhibition of differentiation that I'm observing. And for these studies, I really focused in on the lysosome. And so typically the lysosome has been known to be the sort of the trash can of the cell, where it's the main degradative organelle that has this wide repertoire of acid hydrolases that are capable of digesting macromolecules such as proteins, glycans, lipids, etc. But it's important to note that that's not all the lysosome does. It also has really critical roles in cellular metabolism and also homeostasis. This includes, obviously autophagy, but also plasma membrane repair, cholesterol homeostasis, exosome release, etc. And one of the ways that the lysosome is actually able to perform all these different functions is that it does have a fairly acidic pH. Typically, the ideal range for lysosomal pH is around 4.5. And if there's any sort of defects in lysosomal acidity, it really can have deleterious effects on the ability of the lysosome to perform all these really important functions. So first, in order to determine whether bictegravir is potentially altering lysosomal pH, we use this dye known as Lysotracker red, which is a dye that accumulates within acidic organelles and brightly fluoresces. And when we looked at the number of Lysotracker red puncta within both oligodendrocytes as marked by Gal-C, as well as OPCs as marked with H2B5, we saw a significant reduction in the number of Lysotracker red puncta present within each of those cell types across all 3 doses for Lysotracker and Gal-C positive cells, and decreases of both the medium and high concentration of bictegravir for Lysotracker in H2B5 or OPC cells. So while this is, a nice visual way to say, okay, we are seeing potentially a reduction in these acidic organelles, doesn't actually quantify the changes that we're potentially seeing. So for that, we used the ratiometric dye lysosensor yellow-blue, which enabled us to actually determine what the changes are in lysosomal pH within bictegravir treated cultures. And so here in yellow, I just made a little box that's showing typically what the optimal lysosomal pH is. And we can see that for the medium and high concentrations of bictegravir, we're seeing a significant de-acidification or an increase in lysosomal pH compared to untreated and DMSO treated cultures. So not only are we seeing a reduction in the number of acidic organelles, but we can actually quantify those changes as well. And we are seeing a de-acidification of lysosomes. As I mentioned earlier, the acidity of lysosomes is really critical for their number of their functions, including their ability to degrade cargo. So we next want to determine whether these lysosomes are functional after bictegravir treatment. So for this, we can use a dye known as DQ-BSA red. That is a derivative of BSA that's labeled to such a high degree it actually is self-quenched. And then once it's taken up via endocytosis and eventually fuses with the lysosome in that low pH environment, it essentially generates fluorescent peptide fragments. So it's a nice way to determine whether protease activity is active within the endolysosomes. So looking at Gal-C positive cells, which could be our marker of both immature and mature oligodendrocytes, we can see here in the figures, with Untouched, we have a large number of fluorescent puncta. However, when we look at our low and medium concentration of bictegravir, we see essentially very few brightly positive puncta. And this is just quantified here on the right, where we can see across all 3 concentrations of bictegravir, we're seeing this inhibition of lysosomal protease activity by a decrease in the number of DQ-BSA red positive puncta. We saw the same thing also in H2B5 or OPC positive cells. Again, with looking at Untouched, we can see that we have several brightly fluorescent puncta. However, following bictegravir treatment, we're seeing a significant reduction in the number of DQ-BSA red positive puncta within OPCs. Importantly, we want to determine whether, is this reduction because we're actually rupturing lysosomes, we're actually losing number of lysosomes. And so galectins are a good marker for disruption of lysosomal integrity. Galectin-3 is recruited to damage lysosomal membranes, where it then can coordinate either repair, removal, or replacement process. So here, using leucine methyl ester as a positive control, where we can see a large number of galectin-3 positive puncta located perinuclearly around this DAPI positive inclusion here. However, when we're looking at DMSO and both the low and medium concentration of bictegravir, we're not seeing really any recruitment of galectin-3. So this suggests that bictegravir is not disrupting lysosomal integrity. Additionally, we're also in the process of currently analyzing the total number of lysosomes using LAMP1 in order to verify the findings that we're seeing with galectin-3. So for the last little bit of in vitro work I'm going to mention today, since we're seeing this de-acidification effect caused by bictegravir, we wanted to see if we could potentially re-acidify, if we prevent this this de-acidification, could we potentially prevent this inhibition of maturation? So there are obviously a number of different targets present on the lysosomal membrane that we could approach. For this, this is based on a collaboration with Jonathan Geiger that they had been using targeting TRPML1, which is a cation permeable channel on the lysosomal lumen. It's permeable to calcium, but also zinc and iron as well. And they've shown that this can actually help reacidify lysosomes. So we used the TRPML1 agonist, MLSA1, to investigate these effects. And it's been shown that the accumulation of luminal calcium can actually inhibit hydrogen ion transport and increase lysosomal pH. So we can potentially activate this channel and pump calcium out. We can potentially reacidify lysosomes. So again, using that ratiometric lysosensor yellow-blue, we can see that we're able to reacidify lysosomes across all three concentrations of bictegravir, with both the low and especially with the medium dose being basically rescued back down to that optimal lysosomal pH. At the highest concentration, we did see a significant difference. However, it did not enter into that optimal lysosomal pH range. And this is actually, I think, really nicely verified when we actually looked at this oligodendrocyte differentiation. We can see that we're able to essentially rescue this inhibition of differentiation at both the low and medium doses, where they're returning to DMSO treated levels. However, while we do see a significant difference between vehicle treated high concentration bictegravir and that with MLSA1, it does not return back to untouched levels. So this work is obviously all being conducted in vitro. Another really important area where oligodendrocyte differentiation is really important is for remyelination or myelin turnover. And so we wanted to better understand, does bictegravir impact remyelination capacity in the CNS? And one way that we can model this demyelination and spontaneous remyelination is using this Cooperzone model. So Cooperzone is a copper chelator that, when given ground up in their diet for 5 weeks, we can actually induce really reproducible and reliable demyelination of the corpus callosum as well as the cortex. So here we're taking just C57 black 6 mice, treating them or feeding them either control or 0.2% Cooperzone diet for 5 weeks. And then following that 5-week period, we then take a control and Cooperzone cage for our just verification that we did indeed see a lesion present. And then following the end of the feeding period, we then switch all the animals back to control diet. And it's during this 3-week period that we're going to be orally gavaging animals either with vehicle or bictegravir, which is going to be 3.56 millimolar once a day. And the reason that we chose this 3.56 millimolar dose is that we actually collaborated with the CHOPS Metabolomics Core, where we orally gavaged animals. And then 3 hours post-gavage, we took cheek bleeds and then gave them the plasma so that they could actually measure the concentration of bictegravir in the plasma. What we found was that obviously we know in humans that the plasma max is 13.5 micromolar. And what we found in our mice is that we had an average of about 13.26 micromolar, which is very close to what we're seeing in humans. And then again, this is just sort of the range that we're seeing. So obviously there is some variability, but we were very happy to find that we were pretty close to being able to achieve the average maximum plasma concentration that we see in humans. Then following this 3-week recovery period, we'll assess them for either using fluoromyelin, looking at mature oligodendrocyte numbers, as well as PDGFR alpha positive or OPC positive cells. So we've done 2 cohorts of this experiment, and I just recently got some data using fluoromyelin for the first cohort, so I'm excited to present that today. So we can see using fluoromyelin green, which is a selective dye for myelin, we can see we're looking at the corpus callosum here. we see very nice fluoromyelin green staining in our 5-week control. So these animals were fed control diet for 5 weeks and then sacked. However, when we look at animals that were fed cuprazone for 5 weeks and then sacked, we see really very little fluoromyelin green staining present, suggesting that we did indeed see demyelination following cuprazone. Next, looking at our 5-week plus a 3-week recovery period in these animals. So these are animals that were initially on control diet and then allowed to recover and then either untouched or gavaged with DMSO. And down here is animals that were on cuprazone and then allowed to recover for 3 weeks and then were either untouched or DMSO. What you can see is that in the cuprazone animals, they are able to really nicely remyelinate following cuprazone-induced demyelination. However, when we looked at the animals that were gavaged with bictegravir, what we found was that the cuprazone animals that were had demyelination induced by cuprazone and then were given bictegravir during that remyelination period, they significantly had an attenuation of this remyelination. And so this is just quantified here. So I'm only pointing out just the important statistical comparisons here. So we can see for 5-week control compared to 5-week cuprazone, we obviously have a pretty large induction of demyelination occurring. However, when we look at cuprazone animals that were then allowed to recover and treated with either DMSO or bictegravir, animals that were treated with bictegravir had significant reduction in their ability to remyelinate. However, this bictegravir treatment was not as intense as what we're seeing with the 5-week cuprazone. So they are able to remyelinate a little bit, but it's definitely not to the capacity of either untouched or DMSO treated animals. So just to kind of quickly summarize everything that I talked about today. So we know we have OPCs that are able to differentiate into mature oligodendrocytes. However, if we treat with bictegravir, we increase lysosomal pH. And though I didn't have time to talk about it today, I do have some data suggesting that this de-acidification of lysosomal pH can impact other downstream cellular pathways, potentially by blocking autophagy. And in turn, this leads to a blockade in oligodendrocyte differentiation. However, if we co-treat with MLSA1, we're able to prevent that de-acidification of lysosomes, allow potentially for other downstream pathways to occur, and relieve that blockade on oligodendrocyte differentiation. In terms of the questions that are still, that we're still addressing, still working on, one of the main questions that a really talented undergrad in the lab is looking at is, does bictegravir impact already mature oligodendrocytes? And so she has some data suggesting that already mature oligodendrocytes actually seem to be pretty resistant to the effects of bictegravir. But we're also interested in understanding how it could impact the ability of these oligodendrocytes to myelinate. And so we can use artificial nanofiber cultures that allow us to actually look at how these cells are actually able to interact with these artificial nanofibers and wrap around them. Additionally, does this lysosomal deacidification or this lysosomal stress impact other organelles? And I think, obviously these lysosomes are not in isolation within these OPCs. They are interacting with other organelles, and there's really been a lot of nice work showing the crosstalk between especially lysosomes and mitochondria. And I think, based on Dr. Fields' talk last week, I think there's a lot of avenues where we could pursue looking at this crosstalk between lysosomal dysfunction and mitochondrial dysfunction. Also, if we're potentially able to prevent this effect of a deficit in remyelination in vivo, can we potentially use a lysosomal acidification agent to potentially rescue that? Also, additionally, bictegravir is not only, it's not the only drug given to patients in the clinic. It is given with 2 other NRTIs, such as entercidibine and tenofovir alafenamide. Are there any additive or synergistic effects occurring? Then more of an offshoot of another project that I'm working on, but I'm really interested on how localized calcium release from TRPML1 could impact oligodendrocyte function. So with that, I just want to obviously thank everyone within the Grinspan and Jordan Shudo lab, the CHOPS metabolic core for helping us with the mass spec, and obviously our funding sources. So I know we're a little tight on time, but if we have time for questions, I'd be happy to take them, or I can always answer them in the chat as well.
Thank you, Lindsay. Great talk. Really interesting work. We do, we are right at 12:30, but let's take at least one question. So anyone who has a question, please just unmute yourself.
Lindsay.
That looks like Fatah Kasanchi.
Can I ask a question?
Yeah,
Yeah,
go
you
ahead.
go
Sorry.
Hi, Lindsay. Great talk.
Hi. Thank you.
So a simple question first. How involved is our reactive oxygen species in oligodendrocyte differentiation?
That I'm not, that I'm not super sure about. I think obviously I see Judy's on the call, so she's, but yeah, I mean, maybe Judy has a better idea. I'm not sure.
So if the simple answer is yes, Judy.
Yeah. You can stop oligodendrocyte differentiation cold with too much reactive oxygen species. What happens in preterm babies when they have hypoxia and ischemia and don't get enough oxygen to the brain, and it gets all messed up.
Okay, so Lindsay, what I'm suggesting basically is that you consider the role of the iron store, ferrous iron store in the lysosome, because as you de-acidify, the iron comes out, and of course, Fenton reaction chemistry, you'll get ROS and all that.
Yeah, for sure.
So I think that you might increase your, especially given your background from Olympia's lab and ferritin and iron, it would be a very nice tie between those issues.
For sure. Yeah, and that's definitely something I've been thinking about, because obviously, I mean, the lysosome, has calcium in it, but also is a very important iron store as well. So.
Yeah, great. Thank you.
There is another question in the chat regarding iron, as well as a couple more. So maybe if you could answer those in the chat. I don't want to take time away from our next trainee speaker.
Yeah, of course.
So thank you.